second episode of MyFlora Fridays that's being recorded. I'm not sure if they're going to come out in sequence or not. I'm not sure. But this is my guest, Evan Friedman, who we've been friends with, I think, a couple of years now. Served on a couple of committees together. And he has a very long list of things that he does in the cannabis industry. So, Evan, if you'd like to go ahead and give our audience a little taste of what you have going on and what you do and who you are, that'd be great.
Happily. I am the uh, president of Helma USA and Scientific Cell Company. We're parent companies that is focused on uh, primarily optical spectroscopy measurement of chemistry and how that chemistry changes over time, non-destructively and very, very quickly. And we do this with really any kind of chemistry, be it pharmaceutical or oil and gas or food and beverage manufacturing or chemical manufacturing or paints and pigments. Um, they they all are made up of different molecules, and the molecules are interacting. And when you shine a known quantity of a specific spectrum of light onto a molecule or a bunch of them, uh, the different bonds in those molecules will absorb some of that light. And the different uh, patterns of what gets absorbed tells us what molecules are there and how much of those molecules. And so you can actually control your processes by looking directly at the chemistry as it's occurring. And it turns out that cannabis has thousands of chemicals in this plant. And we can look at the chemicals directly in the plant using a device like the Purple Pro, which is a near infrared spectrometer that measures flower, or you can measure the extraction process where we add tons of value by taking the cannabinoids directly out of the plant and putting them into a format that goes into a cartridge or an edible or a dab, and measuring how efficient that process is and what your inputs are, and really tailoring it so that you get the same predictable output at the end every time. And that's what the pharmaceutical guys are doing. That's why, when you take your Tylenol, it is the same Tylenol that you get every time, no matter if you bought it in a store in New York or in Colorado or someplace else. And consistency and quality and like defining what a thing is and then making sure that you're delivering on that definition and that promise is what we do in every professional industry out there, and what we are striving to do as cannabis joins all of the other drivers of the economy. I said a
Okay.
lot of things.
That yeah. So very chemistry oriented. And I heard you mention the Purple Pro. So I used to have a Gemacert or Gemacert. I don't know how you
That
say it
thing's
correctly.
cool.
It was cool. That company went belly up and my Gemacert became obsolete
Yeah,
overnight.
that sucks.
It sucks. Very, very expensive paperweight at that point.
Yeah, really.
Uh, so I have not used the Purple Pro, but I do. I'm familiar with it. I know what it looks like, and I've seen it in action, but I haven't used it. And from what I hear, it gives very consistent results. Potentially not super accurate, but it depends on what you need it for, right? But is it limited to THC, CBD? Is
Yes.
it's pretty
So
limited, right?
it's it's going to give you total THC, total CBD, moisture, and water activity
Okay.
on dry flower.
Okay. On okay. Okay.
Andre, like you can't take a flower directly off the plant and put it on there. There is too much water in it at
Yep, yep.
that point, and you're not going to get a decent result.
Which is it's easy enough. If you're in flour, you can take it. I bought a cheap food dehydrator from
Yeah,
Amazon. And since you're not measuring terpenes and you're not using it for consumption, you can dry it out enough to test it.
yeah.
And
Since you're reporting the moisture content, you don't even have to get it to a point where you could smoke it. Like you just have to get 50% moisture is probably low enough. When you cut it off the plant, it's like 80% water.
I always wondered, like, okay, so for people who are growing THC type cannabis, I feel like it's pretty accurate. But for I feel like that's what it was made for. But when you switch over to, we were growing high CBD type cannabis. And so our results with at least with the Gemicert were not as accurate because it was
I,
calibrated to measure high THC. And so when you're looking at high CBD, then your THC kind of fluctuates a lot, which is the one that really matters for compliance, right? You want to know, am I under 0.3% total THC? And these little handheld devices, I felt like
so,
they're not great for that. But you make a standard curve and then you figure it out.
and I'd say for compliance, that arbitrary 0.3% THC. Yes, it's a terrible target. It's below the confidence interval. So like um
Hmm.
Gemacert when they were around, I think they said like you could measure that low. What we feel, what my experience in spectroscopy is, it's really great for measuring concentrations between like 1% and 99%. And when you get
Lower.
less or more than that, you're fighting for signal, and it's you start playing games to do things better, but cannabis is a natural product and there's a lot of variation, and
Hmm.
so with the purple, we don't report any number below 2%. Like if the model is predicting a number lower than that, we're not saying it. And that comes out of the results that are reported in a white paper on the model that we've built. There's very strong machine learning models in there, and that's the cool thing. The Gemicert and the Purple, they use the exact same sensor.
Hmm.
The Purple's owner manufactures the sensor, and they were selling them to Gemicert to go into their box too. So it was just how the data was being processed.
Right,
That
right.
was the secret sauce.
Well, and I
And
found that, like, all right, so it would measure the CBD, right? And I found the value of CBD where we would consistently hit in the lab using HPLC, where we would consistently hit under the point 0.3. So I knew to, rather than monitor the THC, I was monitoring the CBD, knowing that when we get to 8%, that's when we need to harvest, right?
yeah,
So I think that, some of these things, I guess my point is that they come with a learning curve. It's not just a plug
they're
and play. You've got to,
right, they're tools.
they're tools. You need to learn your tool. You need to learn its limitations. You need to play with it a lot and
A
figure out
lot.
where how to use it for what you need it for,
A-
right?
And we've got SOPs to offer for different use cases, right?
Hmm.
Because it's like, so okay, maybe you don't like how you're using it for cannabinoids. It's not doing what you want out of it. Fine. The moisture and water activity values are so fantastic for the dry and cure phase,
Mm.
because you can actually track where you are, and you
Hmm.
can make sure that you're staying in that target range, water activity between 0.55 and 0.65. It's like a tape measure, right? So you're just how you point it, where you point it. It's a tool, and
Right.
that's all I'm trying to do is like give people more understanding of what they're working with, and
Yeah.
just you know uh, you can't find things you're not looking for.
And it's, because I've heard people say, oh, the Purple Pro, it sucks. And I know that the Purple Pro 5 years ago was not as good as it is today. But also, these tools are not meant to be a replacement for things like HPLC and GC, which are, a lot more expensive and a lot more
Yeah,
sensitive. But
orders of magnitude more expensive.
right, they're not substitutes. They're
Supplements.
supplements, right, to help you kind of guide your grow and where you want to be. Um, no, I like, I would like to have one of everything. I think
Yeah, right,
is it
exactly.
is it John McKay who has like
Ah,
all of the things and
yes.
brings them all to the conferences and shows everybody how they each work. And he's a fan of all of them. He's like they're all different.
Yeah,
They all serve different purposes. You need to know what your goals are, and then learn how to use this instrument to your best benefit.
for sure.
So so that's
My favorite thing about the Purple Pro is like you can measure every bowl you smoke because it's non-destructive. You're just shining light on the flower.
right.
Like when once you're done, you've lost nothing. You can measure it over and over again. You or smoke it, and
And you
that's
can
fun.
you can measure something and then send it to the lab and see how close your results
I,
are. And then you can get an idea of how far off is my instrument versus their instrument so that then you can gauge like if I'm right here, then
yep.
it's going to be good on this end.
And
Uh,
what's cool is when we build the models, we sit in the lab. They're doing the same testing that course that they'd be doing for um, compliance. It is
mhm,
compliance testing.
mhm,
And we just before they do the sample prep to run it on the HPLC, we run it on 8 different devices. And
mhm,
so we have a data point. So we've captured the variance between devices too as we build up the model to
mhm.
try and really make that prediction as robust as possible.
I like that. I'm a fan of multiple data points.
I mean, we're a spectroscopy company at our
Yeah.
core.
Yeah.
Both Purple and their ownership and Helma and Scientific Cell, like our loyalty is to the measurement, the data, the purpose of it. And like if you don't do it, if you don't set up your experiment right, the stuff you get at the end is not telling you what you were looking for.
Right. And so really like your customer base it's going to be cultivators, correct? And what
A lot of cultivators,
are they
breeders,
shooting
right? Think
for?
about pheno hunting with a device like this and culling the ones that aren't performing. Then it doesn't even have to be that accurate. It just you plant to plant, measurement to measurement. As long as you're finding the ones that are lower and higher, you can guide your pheno hunting process too,
Only if you're hunting for the highest THC, which
or the highest CBD,
well,
or you're looking for the ones that are ratioed in the batch, and you can see that.
that's true. That's true. Touche.
It's
But yeah, no, I have had a couple of conversations about uh, you know, many cultivators are selecting for the highest yields and the highest THC.
yes.
And
And are they washers?
yeah, and to the detriment of flavor profiles, aroma profiles, that there's
Medicinal effects. I don't think the aroma profiles have suffered that badly. I think there's been a lot of really cool novelty there. But the chemical diversity, right? Like it all starts from olivotolic acid, right? And then CBGA and then other cannabinoids. And it's like if you shove it all down the THC pathway, it's not going down these other pathways.
right.
And that I think that's why people are like, this isn't the same high that we used to get 20 years ago. because it's
Right.
like everybody has selected really hard on this one biosynthetic pathway.
Which, I mean, we've all heard people saying that I don't like the weed today that I can get because it gives me anxiety, or, there's lots of people complaining
Or
about,
CHS stuff.
yeah, negative effects. And this isn't, where's the giggly stuff that I used to smoke back in the, 70s, 80s, 90s? I can't find it anymore. It's because I don't have any proof, but I'm pretty sure it's because we've been,
Well,
selecting for and breeding for higher and higher THC, and it's like a teeter-totter, right? You got all of your weight on THC, you're losing,
it's
all of these other beneficials.
absolutely and it's actually I think it's like quantum uncertainty. It's Schrodinger's THC because we started measuring it, right? Like we started sending
Mm-hmm.
the plant to laboratories and measuring these numbers and saying THC, this is the one that gets you high. And so if we're looking for it, we're now directly influencing what's happening and we're picking for it.
And
And it's
it's really the
accelerated
only,
through legalization.
in a lot of states, that THC number drives the price.
Yep.
So the things with the highest THC are put on the highest shelf are called their premium or platinum or whatever.
Sets the floor of acceptability,
Right.
right?
And so that in a consumer's mind equates to a quality product because in
Yep.
everything else, the higher the price, the better the quality. That's how it's supposed to be. And there is usually in most of the markets, there's no other quantifiable way for a consumer to make a decision on whether they think it's a quality product or not, because you can't see inside the packaging, you can't smell, you can't touch, you can't look. Like all that they
No,
have to
like
go off of a lot of the time is that THC number, and it's the regulatory agencies that have put such an emphasis on it. Like we need to know the THC. Well, you can only have 10% THC in any edible, otherwise you're gonna die.
well,
And
the edible is actually the most interesting case. I think that's the one where you do need to know the
well,
THC.
I think you need. I mean, no, yeah, you're
The
right. It's
the
like
flower, I don't care, right? Like it's a plant that THC is going to be mediated by what else is in the plant, and but like how much THC is in an edible? That's going to go through your first pass metabolism. You're not consuming
right, and
THC.
they're
You're
different
consuming 11
molecules.
hydroxy, right?
Right, they're different molecules, and you can. I think you can better titrate your dosing when you're consuming
Smoking.
flour because
Yeah.
you take a hit, you wait a couple of minutes, and you know what's going to happen to you. With edibles, you take one edible, you're like,
It could be 2 hours before anything happens.
and it could be based on what you ate, whether you like, there's so many,
So, I say this to a lot of people. Maybe I haven't thought it through enough, but like flour to me is produce. And it should be treated like produce in every single way, from a regulatory standpoint, but also from like a consumer quality standpoint, right? Like if it were strawberries,
hmm
they would be refrigerated within an hour of harvest at the absolute maximum
and
and
wash.
uh, and washed. Why? That's a sticky wicket. We can talk about that one, but into the cold chain, and that cold chain held to the grocery store and the consumer at the end the whole way.
Right.
Preserving.
And if they're moldy on the shelf,
Don't buy it.
don't buy it, and the store should pull it. Like, or
Right. And if you can, I feel embarrassed about this, but I have taken a thing of strawberries back to the store that I bought the day before and been like, no, these are moldy. I want
right.
a different one, or give me my money back.
I would do that too if I wasn't so lazy.
And yeah, right. Like I'm
And no, if they were if it was a $30 tub of strawberries and they were
yep,
moldy in 2 days, I'm getting my
yeah,
$30
of
back.
course.
If it's
right?
$2, I'm not going to worry about it.
And so like, you talk about consumer quality of cannabis, it should be fresh. It should not, it doesn't need really all of this testing because it's produce that you're going to light on fire. And if you are concerned about uncleanliness and negative health effects, you should be consuming medical cannabis that is held to the higher standard. And I think like we should treat it like the agricultural produce that it is and like make sure that people uh, are getting fresh flowers.
Hmm. Hmm. No, I can get behind that. Um, oh, I mentioned washing, and I know,
Washing, yes. Clean washing in terms of cleaning.
yes. So, when I was working at the farm I was working on, we did wash our flour. We were growing in hoop houses. So not a completely sanitary sealed, room. Uh,
Agriculture, life is not really sanitary. Like
right.
trying
And
to
he
maintain that environment is really hard.
and before they grew CBD, they grew produce. They grew tomatoes and all kinds of lettuce and kale and things like that. And they had a 3 compartment sink that they would wash and use peracetic acid, which doesn't necessarily wash the produce, but it does, but it also sanitizes the water that you're washing the produce in. So it makes the water sterile because water is not sterile either. There's
No.
stuff in the water too. So even when you're washing your produce, there's stuff in the water that can make something grow on your whatever it is. So you use something like peracetic acid that's going to kill everything in the water as well as whatever is on your produce to make sure that it comes out as sterile as possible with everything that could grow is then dead, right? And so I know, so I've washed buds before. I'm a big fan of it. I've seen what comes off of flower when it's not grown in a sterile environment, and it's nasty. And I know that there's lots of people who are like, oh my God, like you're gonna wash off all the trichomes, you're just gonna create more mold. Um, my argument to that would be it's a plant. It doesn't the trichomes don't just fall off. Like there's rain. It's kind
Yeah.
of like akin to like the hair
Well,
on your body. Like it doesn't just fall off if you
right.
rub it.
And again, right, the like that flower is like 80% water when you take it directly off the plant. So like
Mhm.
if you're washing it at harvest, it's you're it's all supple, right? Like it's full of water. It's when you start drying it out that those trichomes become brittle, that you can lose terpenes or their heads or whatever. Like when it's fresh, like yeah, you can't wash it vigorously, but I don't see
No, it's a
why.
gentle cycle. It's not a
Yeah, right. You're not making hash, right? Like if you want to make hash, that's a different story, and we should talk about that.
Yeah, yeah. So, so I like the way that you frame that as flour should be treated as produce, essentially.
And then
And
you start processing it, and you're into, you know, chemical manufacturing and consumer packaged goods. And that is a different thing than produce.
mhm. And so, another thought that popped into my mind is this idea of irradiation. Um,
Mm.
people have flour that fails, and in order to get it to pass, they do things like irradiation, which kills the microbes, and then they send it back in, and then it passes, which I think is a terrible procedure. It just shouldn't happen, right? You wouldn't wash a moldy strawberry and then put it back on the shelf and say, no, it's good to go. Right. So back to your idea of produce, but I think there in crop production there are like mitigation steps. Like you might irradiate or do some UV light treatments during the growth phase to sort of have kill steps along the way.
When it's alive,
Hmm.
I feel fine about that.
Hmm.
I don't think that's an issue at all. I think it's like that, then it is a dynamic living organism. And yeah, maybe you're going to have some effect on the outside of it. But like, I, I actually think the best mitigation step is just growing it in healthy living soil.
Hmm.
I think if you've got soil that is rich with the right kind of life and it, you have an ecosystem and it's going to be fine.
So then hydroponics and aeroponics,
Not my favorite.
not your favorite.
I don't think you get the richness in any biosynthetic production that way. I think that is, hydroponics is like something that came out of prohibition essentially. It was like, how can I grow more plants in my limited space
Hmm.
without being caught hauling, a literal ton of dirt down into my basement.
Well, I mean, but there's other crop industries that do use hydroponics, right? Because you can grow a lot very quickly, right? So there's huge
Yeah,
lettuce farms that use hydroponics.
because you turn very fast,
Yes, it
right?
grows very fast. You get big crops. It's very clean if you do it right.
But I mean, how are you just going to grow an insane number of really small cannabis plants?
Oh,
Like,
no, you can grow them to full size. Same thing with
yeah,
aeroponics.
but how quick? But how quick is that going to be? Like, you're already losing the pace that hydroponics gives you with lettuce or strawberries or something like that.
You can shave off a few weeks of your
Yeah,
growth, yeah, and still get the
you
same
know
size,
better
which
than
is,
me for sure.
yeah, which is, I mean, there are a couple of aeroponic companies, a lot of hydroponic companies, but aeroponics you can definitely grow quicker.
But I think
Aquaponics is,
those plants are more stressed without the support system. I don't like feeding plants salts either. I
yeah,
think,
yeah.
right, like living environment provides the richest profile. It gives the plant the most opportunity to express its potential.
Right. I agree. And, so we're trading something you want more harvest per year. So let's say you can squeeze in a whole nother cycle at the cost of what? Right? So, you're growing faster, but then
Is it product quality? Is
the
it the
plant
cost
doesn't
of
have the
product
time
quality?
to the plant doesn't have the time to develop all of the things that it
Yeah.
could develop in the normal time frame. So, you know, we've talked about, people chasing THC and chasing yields. Okay, so you get the same amount of yield, you might get the same amount of THC,
chasing
but
chasing flowering time, chasing
yeah, but then now you've got all this flower that really is devoid of
character.
character and complexity.
So my friend Adam Jacques was telling me that for the last few years he's been growing um, in living soil in beds inside with cover crop,
Mhm.
and he's on 12-12 continuously.
Hmm.
And so these plants just get to the size that they get and start flowering. And he gets 5 yields a year out of this.
Is he growing autos then?
Sometimes it's autos, but no,
So he
he's
doesn't flip it. It's just,
no.
I like some of these folks that are messing with stuff.
Oh,
It's,
he's a brilliant breeder. He's got cool experiments going for
yeah,
sure.
Colin Clark does some stuff and he's going to be on MyFlora Fridays coming up soon. But he messes with stuff and has found, the light dark interruption does it, things he just messing with plants.
Yeah, right. Like, I mean, are they going to complain?
I mean, maybe.
Yeah, you'll learn something though.
Yeah, yeah. And sharing that data when you are messing with plants can be so valuable. Um, and unfortunately, with cannabis being illegal for so long, there hasn't been a lot of opportunities
Yeah.
for growers to share this information. There's a ton of legacy growers that did a lot of messing with plants, and either, committed it to their steel trap memory or, some of them wrote it down, but they weren't able to share what they were doing because jail.
Right, right. Like only the crazy ones wrote it down because like evidence to put you in jail.
Right, right, right. Uh,
But Adam's a really interesting guy. Um, he's was developing CBD medicinal strains before the Stanley brothers even. He's got a couple strains, Frank's Gift, just real dedicated medicinal strains that he developed in the early 2000s.
mhm.
Um, how he's in Oregon these days, but
Okay, well, that's a good place to be.
yeah.
Uh, and there's a lot of talk about microbes and like, all right, we're testing for this. Is it really harmful to humans? Like eating versus lighting on fire and inhaling is two different things.
Definitely.
Spores,
Or
dead.
how about dry herb vaporizing, where you're inhaling but not even combusting?
Yeah, yeah. And you've killed your microbes, but then the toxins could be left. So
Yeah,
if you're doing a plating assay, the microbes don't grow because they're dead. That doesn't mean the toxins are gone, right? And then uh,
there's a
some of
lot of toxins on that potential list too, and they have some nasty symptoms.
yeah, yeah. Well, and um some but some of the tests people are saying are, I don't want to say stupid, but maybe unnecessary
Yeah.
because they were born out of the food industry.
Yep.
Uh, and so unless you're ingesting these things,
Well,
it's
right.
a different sort of situation of consumption. So we need data on
That
consumption methods and impacts, but there's a part of an ethics problem there too. We can't be having people smoke pot to see if they get sick or not.
Well, I think, well,
I mean,
we've
they
been
already
going
are.
through the largest uncontrolled study for quite a while
That's
already,
true.
but
That's true.
um, so the ethics thing is more of a thought experiment, I guess. But I think that's again where my distinction of agricultural produce or processed goods is becomes quite useful because it's like if it's a processed good, you can study it in controlled conditions, but an agricultural product is going to be different all the time.
Yeah. Like
And
like you buy a Granny Smith apple, what's the sugar content? I don't know. Like where was it grown? Are they
I
good apples from Tasmania or were they grown in, I don't know,
do you
somewhere.
know what your preferred target sugar content even is? Like, I mean, I could take a Purple Pro and with a little bit of effort train it to measure sugar content in apples. Like that is like that hardware could do that with some effort. Um, Not an insignificant amount of effort, but really very very achievable.
But could be done.
Very achievable with that existing piece of hardware. But like now I'm telling you the sugar content or the bricks value of your apple. What does that mean to you? Right? Like just like I'm telling you the THC content. What the fuck does that mean to you?
Well, there should be like a standard range because I don't know, like Honeycrisp apples have been probably my favorite apples for years. There was a few years where they just sucked. Like no matter where you got them from, they just sucked. And it took like 5 years. I didn't buy them because they sucked.
I
They're back now. I don't know what happened to the
Yeah,
Honeycrisp
right. That's
apple.
a really interesting question that somebody has an answer to.
Oh, I'm sure. All right, so I want to move on.
Okay.
You are involved in the Northeast Pheno Cup.
Oh man, that thing
Uh,
is so cool.
so tell us about that, because I have questions.
A number of years ago, I got put in touch with Scott Odette, who is the founder of this thing. And he had you know, was involved with all of these breeders and the medical market. He comes from the caretaker system in Massachusetts.
Hmm.
And the breeders all wanted feedback on their seeds and instead of just doing pheno hunting themselves, they would uh, they'd share the seeds, they'd do sort of distributed stuff, see what it's like under different conditions, and somehow that idea developed into a competition. And so every year run three categories, although I uh, I've been trying to get rid of the categories, which are, indica, sativa, and hybrid. Everything's a hybrid, hybrid, hybrid these days. So that those categories don't make that much sense. But like we do three seeds, and everybody that enters grows the same seed in the category. And so it's this big distributed pheno hunt. And then we do judging just like any other cup.
Mm.
Um, you know,
So it's a
I,
competition for skill and also sort of a measure of diversity. So you're not looking for like they all start with the exact same thing.
right. Not
Not
everybody's
like the
getting the
grow
same
up.
cut. Everybody's getting the same grab bag potential. And so it's at least 10 seeds per per strain.
Okay.
Per category, right? Because they're sibling seeds, right?
Right.
And there's a lot of diversity in there.
Right.
It's not going to be any more stable than an F1.
So though for those people who are not or who are interested don't really understand this, like it's the difference between, your parents they have 10 kids. You all have similarities, but you're going to be very, very different, right?
Yeah,
You're going to be different heights, different potentially eye colors, depending on your parents' genetics. But, it's a mix of 50-50 of each of the parents. And so, or like if you adopt two dogs from Greece where they're mutts, and then you have a litter of puppies, you're going to have variation among those puppies. They're not going to look like
dream variation.
right. So
A lot of potential there.
you've got variation in these seeds. They came from the same seed lot. They're essentially all brother, sister,
Yeah,
hopefully
they
sister.
have they have the same parents, but there's a lot of potential there.
Hmm.
And um, the idea is that the winner in each category um, gets uh, the breeder has the right to buy the winning cut back, and the winner gets the right to name the strain.
So
Um,
it's like a crowdfunding pheno hunt, sort of. Or
I Yeah, it's definitely got this collectivism vein in
well,
it, and
I like that because pheno hunting is a very intensive process, right? For one person to do. You've got to grow up potentially, hundreds, if not thousands of plants. Then you have to assess each of those plants. Then you have to pick one or however many of those plants to be your winner. But you're spending all of your time, your, resources, your money on finding that winner. Whereas if you have multiple growers doing the same process, like it just takes the burden, it lifts some of the burden off of the breeder, right?
yeah,
I like
yeah.
that. That's pretty cool.
It's right. Like the one breeder has their setup. They've dialed in how they like their lighting, how they like
Mm-hmm.
their is it soil, is it hydroponic, whatever. They've got their growing style. And
Mm-hmm.
so when they're pheno hunting, they're
It's
doing it either in their growing style or in their set pheno hunting, but it's all the one way.
yeah,
This way, everybody's style gets to try their hand at it and see what can come out too. So I uh,
not
you
only genetic diversity but environmental diversity.
yeah, right, like there's nature and nurture.
Yeah.
And the interplay between them is the secret sauce.
Yep. So you do your own podcast. So I'm
Yeah.
learning. This is, like I said, this is only my second time doing this. I have been on multiple. I've been on your podcast, and we did the Dank Hour, which was a panel of people, and we would have a
Yeah, that
guest
one's cool.
on. But this is my first time doing like my solo thing. But I figure, I speak to cannabis folks every day. We don't record the sessions, but we might as well, right?
I
So,
mean, that is the origin story of my podcast. I was with Brian Fields and Kellen Finney at the Emerald Conference in 2019, must be like a long time ago, and we had just gotten out of like the third really fascinating conversation in a row, and I was
mhm.
like, we should be recording this.
Yep.
And that turned into the two of them starting their podcast, The Dime, that puts out an episode every week. Since then, since like November of 2019, they have put out an episode every week. It's incredible.
Wow.
I was not that ambitious. I aspire to put out an episode every month. Sometimes that happens, sometimes it does not. But fascinating conversations with people all the time. Really love getting into it. And yeah, just hit record. How hard is that?
Talking about our podcasts, we've met each other a couple times at various conferences. I feel like we
Yes.
should be carrying around microphones, like talking. In one of my plagues, one of my toxic traits, or whatever, is that I am not very good at asking people questions. So this is like a new thing to me. Like
Oh, that's great.
I'll have a whole conversation with somebody, and not once will I ask them anything about themselves because they've been asking me like I just don't. Boyfriend, perfect example. He'll come home and say, "How was your day?" And I'll say, "Oh, it was really good, blah 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 blah." And then he'll say, "My day was
How was
good
my
too."
day?
I'm like,
Yes,
"Oh, I'm sorry, I forgot."
I have um I have encountered this criticism before.
The same criticism that you don't ask questions or
I,
seem like you can't share.
I live in my own sphere at times, I suppose.
It's not for lack of caring, and
No, not at all.
I do after the fact. I'm like, oh shit, I should have asked them about this or whatever. I just don't. It's yeah. Anyway, back to cannabis. So we're talking about the Purple Pro.
Yeah,
You also work with Vern Bio. They have a couple of different tests. What sex testing?
sex testing.
Sex testing. So I know it exists for a reason. We don't want, there are people who like to grow regular seeds, which would be a 50-50.
I would encourage people to grow non-feminized seeds.
Okay. And why do you,
Feminization is the the product of applying a stress to the plant, which means that there is a good chance that feminized seeds are potentially weaker in a number of ways and more susceptible to any number of afflictions or diseases.
so you can produce male structures through stress, but what about applying hormones? That's not necessarily a stress. It's like you are blocking certain hormone pathways, and therefore the plant sort of switches, not unlike with hormone therapy for transgender humans.
So you're talking about growing plants with Y chromosomes, but never allowing them to express male traits.
Other way around, growing female. So the thing about male plants is that you can't see the traits that they offer to the female flower. So you can get an X chromosome from your father that will contribute to the femaleness of,
Of course.
right? But you can't see the because they don't. produce the same female flowers that have the cannabinoids and the terpenes and all that. Like you
It's
don't
really
really
hard to
know.
hunt the males.
So you have two females that you really like their traits. You can reverse sex one of those females using hormones to present with male structures that you can then use the pollen from that female plant to pollinate that other female plant. Now you got a feminized seed. But and some people just if a plant has a stress, it produces some pollen. You're like, oh yeah, I'll just do that. That's not how you should be breeding. Um, I know a lot of breeders who put their females that they're going to reverse sex through a stress test to make sure that they're not going to crumble under the stress. They're gonna hold their, like Queen Elizabeth II, I'm not doing anything, I'm not going anywhere. And then those are the two female parents that you would then use to make a feminized seed, right? So in the case that
Okay.
you're breeding
That's
correctly.
interesting, right? Because I've every time I've encountered this discussion about reversing the females,
Hmm.
um, it was for the purpose of additional breeding and reinforcing traits, not in producing feminized seed to like sell to the market.
Oh,
But what you said obviously makes perfect sense. It's just, most people I see, most feminized seed uh, comes from like spraying silver colloid onto plants and stressing them out so that the seeds come out female.
well, the colloidal silver, the silver or silver thiosulfate is a hormone that blocks ethylene production that then the female can produce those male structures. So it's just a hormone blocker. It's not stressing the test. It's not stressing the plant out in the same sense where there's like light stress and then it produces some male structures or it goes undergo, the plant gets too old and then it's like, oh, I need to make a seed. So they're like two different things.
Hmm.
If you've got rhodolization going on, which is where the female gets old and she's like coming to the end of her life and she's like, I need to make a seed so that I can try again next year. That's, I don't know if that's good or bad that she's waited that long to go ahead and try to set seed or, some kind of drought stress. And then all of a sudden you've got, pollen sacs forming or there's a light
Uh-huh.
leak and you've got pollen. That's a low tolerance for stress, which is what you don't want to carry through a line, right?
Right.
If you do a stress test and your plants don't do that, then you've got a plant with a higher tolerance for stress, and that is something that you want to carry through to your next generation. And those are good candidates for the reverse sexing with something like silver thiosulfate or I forget what I think you can use like gibberellic acid maybe. I don't know. There's a couple different ways you can do it. You just spray the site where you want the male flowers, and then you can collect the pollen and go ahead and pollinate your other plant,
Uh-huh.
or itself if you want.
Yeah, right. So that's I haven't thought about it that deeply. I am not actively involved in the process myself.
Right.
Um, but so I nevertheless, people do want to grow regular seeds, right?
Back to
Or
the point.
people have regular seeds, right?
Yeah.
Who cares what you want? Sometimes those are the seeds you have, correct?
Yeah.
And so the sex testing at least gives you the opportunity that to pull the genetic males, the ones with Y chromosomes,
Hmm.
out before you've invested any resource into them.
Hmm.
So the samples that you can take that you need are really tiny. And so if you germinate that seed, as soon as you get the first node after those initial cotillion leaves,
Cotyledons.
cotillion, that's not the right word.
Cotyledons.
Cotillion, yeah.
Cotyledons.
Those coddle leaves, uh,
Cotyledons.
cotyledons,
Yeah.
cotyledons.
Cotyledons.
Yeah. the next node up, you take like a small, you take a leaf if you're going to send it in or if you're going to run them on site, just a small punch out of the leaf. And that's enough DNA to like tell. So, I mean, you can know it's a male in week, what, two, three?
I mean, they should. Well, and so something that I think about is okay, so you've got a population, let's say of 1,000 seeds that you've germinated, they're regular seeds. In a perfect world, you're going to get 500 males, 500 females. You pull all those males, golden. But there's a chance that of those female plants,
You've
some of
got
them are
herms.
still going to
Yes.
express male
Yes.
characteristics.
A hundred
There
percent.
might be one, there might be two, there might be none. In my experience from feminized seed, it's usually about one in every 2,000 that expresses male characteristics, not because there's a Y chromosome, but because sex is not only the result of chromosomes, it's also
Yep.
hormones and receptors that fall, that,
Yep.
and
And
so I don't know. Uh,
and depending on strain, right? Like that it could be
yeah,
far more often, right? Like there are some strains that just herm, like
yeah, because they have a low stress tolerance or
right.
whatever the case may be, or maybe you got a light leak in your hoop
Or
house or
or
something.
or you are closer to the original landrace genetics and those plants, plants evolve to mate and reproduce, right?
That's
Like
that
life
is their
evolves
sole purpose in life is
is
to
to reproduce.
reproduce. I mean, that's everything's.
Yeah, right. If it's alive, that's its purpose,
That's
right?
its purpose.
And so, you think about some of these cannabis plants that grow on mountains in Nepal or something like that, and they're probably far more likely to herm because maybe it's one plant growing in an outcropping and like it wants to reproduce. And
I
so
mean, yes, if it's a solo plant, it has the mechanism to be able to set a seed before it dies. Rotalization is that process generally, but I I would disagree because most wild growing populations are going to have males and females, and so that reproductive uh inability is not there. Like in our cultivation systems, we are on purpose
yeah,
removing the males, and so I feel
yes.
like a lot of the females, because we are keeping them apart,
We've driven them to this.
I think we have, and I think there's more of a tendency to get herms in your grow because we're not letting them do what they want to do, and females do what females want.
Right. The plants want to reproduce.
They do,
It's
do.
their only function, essentially.
And when there's no pollen available, they're going to get bigger, they're going to get fatter, they're going to get
Yeah.
stickier. They're going to do everything they can to catch any pollen in the area.
And the males, if there's no females around, are going to get as big as they can and release as much pollen as they possibly can because it needs to go as far away as possible.
Because it,
And that's what we see in a lot of the cities in the East that sometime in the 50s there was government guidance about what to plant in cities to like tree wise. And the guidance was that they would only plant male trees in cities because it would make for no fruit to and pits and stuff to clean up
messy,
off the
messy.
streets. It's messy. It's a social burden, whatever. And and then there was some blight, and all these urban trees died, and so they replanted off of all this guidance. And you live in New Jersey now. What is it like in the spring? The green coat that you find on your car. Just the trees produce so much more pollen because they're looking for somebody to pollinate. And
Yeah.
the plants want to reproduce. It's just
the
It's
pollen
hard to
indicator.
ascribe motivation to plants, but like that's the one.
Right, right. And I was reading something by Ernest Small uh about the male plants. Not only, I mean, cannabis is a prolific pollinator. Just, you know, what people call pollen sacs are actually the male unopened flowers, the male buds of the male flowers, right? Once that opens and starts producing pollen, 350,000 grains of pollen per flower, per one of those little pollen sacs, right? So you think about how small that is, and if you've ever seen a male flower or a male plant in full flower, there are thousands of those. So that times 350,000 pollen grains, and they don't stop. Like they don't just
No, those flowers
stop.
open
They
and
open,
release daily.
they release, and each individual flower will eventually wilt and die, but more pollen sacs will, more flowers will, it'll keep going. And to think about how,
Yeah, for like
and this was
a
a
month and a half, probably.
this was based on a pollen count, I think in 1996 in the United States when cannabis was still illegal. So this is wild, growing ditchweed that we were getting this pollen count from. Um, and it was something like it was making up something like thir 30 or 60% of the pollen count was cannabis pollen. And I'm like, how? Like, that's so much cannabis pollen for a place where it's not even legal to grow.
Yeah, right. Right. And so most of the growing is inside,
Well,
right?
in
Like,
1996, no one was growing. It was either like
yeah, right. Even
weed or
even the
whatever, but
illicit growing was probably inside.
right, and not males. And not
Not
males,
males.
right?
Not
So,
males.
and I don't know, somebody made a good point that, uh, cannabis pollen and hop pollen are almost identical. However, same thing with hops. Like, we don't grow male hops. There are some wild hops out there, males included, but that's still like a huge amount of Cannabaceae pollen floating around in
Yeah,
the air.
right, right. I mean, there's only two plants in this genus, right?
No, there's some more. Celtis, Trema, a couple more. They don't matter. I only like
Yeah, it's a beer,
party plants.
beer and weed.
Right. But I don't know the structure of their pollen, and they're trees. But I don't think they're like everywhere.
They're
But anyway,
trees.
nonetheless, I think
They're trees. They're not like vines or bushes.
right, and I think it's super interesting that there would be that much pollen in the air making up the pollen count. Total,
Yeah, right. I, I'd be interested to see a follow-up study.
right? Now that it's legal and people are growing hemp everywhere.
Yeah, right. There's all this hemp growing.
Yeah. So yeah, somebody needs to do that study. Hey, everybody, somebody do that study. Um, so I guess knowing who you are and what you do, Evan, I think we should finish up talking about the importance of science, standards, and education in the cannabis industry and the role that you play in each of those things.
Okay, let's
This should be short, right?
let's. Oh yeah, let's do it backwards because I think education is the one that deserves the most time and needs it. There's lots of people working on standards, lots of them. Uh,
And by standards, we mean
like agreed upon uh procedures or metrics or um minimum quality expectations uh, of be it products or procedures or labels or um,
facility
yeah, right, the manufacturing components or
recalls
I mean, regulations are standards too, or how the measurements are conducted or how the samples are taken. They're all they
are
all
all
could be
standards.
standardized.
So, and by they all could be standardized, are you talking about within each state program or among all of the states and make it all a cohesive unit where every state does it the same way, we're all on the same page?
We live in a lovely country where we have a state system and a federal system, um, for most things. Uh, for cannabis, that is not what is currently occurring. Um, the way the regulations have proceeded has been a lot of copy paste from one state to the next as they've gone through legalization and regulation development, and some backwards revision, but not much, which is not surprising. Actually, in most industries, the only time the regulators change things are when they get sued. So, like, we act like it's crazy that the regulators are getting sued, and that's the only way to change things here. But that actually seems to be the case in most industries. The
But I have
government,
to say, in a lot of cases, the regulators have one job, that is to regulate. And in a lot of cases, they're not really regulating. They spent,
are we talking cannabis now?
oh yeah, they
Because
spent a lot of
yes.
time making up all the rules, and then they just said, "Go play the game. Here's
Yeah,
your
we,
rule
we
book."
we designed this game. We didn't think about what happens when it's actually being played,
Right.
or how people might
Change the rules
use the rules, change the rules. I mean, like,
when the
talk about
regulators
what
are not
state.
looking, which is most of the time.
It wasn't copy paste between these states. It was copy pasta. Like they made little changes that had huge impacts in each of these changing jurisdictions. And so, I mean, a unified standard across in terms of what, you know, I think every MSO would love a unified standard in terms of packaging. It would make them actually be able to achieve economies of scale in buying packaging. But instead, all these operators have to buy packaging by state so that they can, meet each individual state's labeling requirements. And, when New York opened, the packaging had to be bland, like really bland.
Mm.
And then the regulators got sued and lost. And now the regulations are open again for what the packaging should be like. What? I mean, this is over regulation to begin with, right? Like, I, setting standards is really important. It's important that we're all measuring the same thing. And so AOAC is the Organization of Analytical Chemists and has been involved in setting the requirements of what measurements should be and then naming acceptable official methods that meet those requirements for lots of industries for well over 100 years, and has a charter from the US government, and took on the challenge of helping set some standards for the analytical testing of cannabis. And some of the states, in their regulations or in the laws that point towards the regulations, actually name that the methods being used in the compliance laboratories have to meet AOAC standards, um, which could mean a lot of things. And there, the interpretation of laws is an entire field, isn't it?
Yeah.
And then there's organizations like ASTM, which also for well over 100 years has been setting standards, often more engineering type standards, but uh, they've got a committee D37 that has been trying to set all sorts of standards for the cannabis industry, be it packaging or labeling, trying to just help be a voice that can create a point of alignment. Because when everything's different, not only is it difficult for us in the industry that are used to these things and just frustrated with the differences, it becomes impossible for the typical person that just wants to go into a store and get some flour. I, you know, I, I think back to the first time I went into a licensed dispensary, and it was like an Apple store, but they had a million SKUs on the shelf, and I couldn't tell the difference between any of them. And I'm like, what is going on here? And why? Why should I pick this one or that one? And I think that's why, you know, THC has become this like metric, because the
It's
regulations kind of said this thing that you should be using to decide on your eyes and your nose is going to be obscured, and so here's this number we're going to give you, and that became the conversation.
mhm.
And it's
And
just
no
like,
matter where you go, you can see that number. No matter which state you're in, no matter what packaging there is,
and
that number is going to be on that product.
and
There's no
so
state that doesn't have it.
it's essentially the only unified standard there is,
That's true.
which is kind of psychotic because it actually predicts very little about your experience with that flower.
Right.
There is a wonderful paper from a number of years ago now called The Nose Knows,
Uh-huh.
by A.D. Ray and Jeremy Plum. I think
Yep.
Ethan Russo's on there. And they did this really cool study with a grower's cup, and they had really robust data gathering from the judging panel and analytical data on all the results and everything. And they showed there was absolutely zero correlation between the individual's experience with the flower and the THC value.
Right.
Um, the result really showed that the only thing that predicted experience with a product was the person's subjective assessment of the flower by smell
Yep.
in advance of smoking it.
And they even tested terpenes, which had very little impact
Yes.
on the consumer's
Now,
experience.
now I'm very curious. This is before the the studies from abstracts came out.
Yeah.
And so there was no even thought about testing for sulfur compounds. But I'm curious what a data set like this might reveal if the sulfur compounds were in there too, because they have very low concentrations, but very large impact on the aroma. And I think people react to them very differently.
Oh, I'm sure Abstracts is working on it. It's hard to get a large group of people together and then to test all that flour, that it's
It's a lot of
very
money.
money, very time consuming. You've got all
Yep.
that data to analyze. So I wouldn't be surprised if, Abstracts and, our friends Twinkle and Ian and those guys are TJ are all working on that question right now because the volatile sulfur compounds and some of the other flavorants seem to have a lot more impact than the things that we've been looking at, which is THC and terpenes.
Yeah, right. Exactly. It's like, oh, these tests you must do. We're measuring these things that have almost no impact.
Right, right.
It's
Yeah,
like the aspergillus testing. We're testing for this contaminant. It's everywhere. It's in it. Like asking people for it to not be there is actually kind of crazy.
right.
And
It's like
it,
testing for dust. Like,
yeah, right, right. It's just test it for total carbon content. Like
all right. So now we know what standards are and what
so
your
that's
definition of
standards, and I do
standard.
a bunch of this. I work with AOAC. I work. I Many of the people we're friendly with work on these ASTM committees, and I also involved with ACS CAN. That's the cannabis subdivision of the American Chemical Society, where We're really gathering together all the chemists and a bunch of other chemistry related scientists, um, and focused on cannabis in a really rigorous way. The American Chemical Society is 125 years old, more, and has divisions for rubber and medicine and cellulose and
The cannabis division is relatively new, right?
yes, in fact, it isn't a full-fledged division of its own. It is a subdivision of the Chemical Health and Safety Division still, but we are in the process of coming into our own as a full division.
I submitted an abstract in 2019 uh about THC inflation.
In 2019 already. Oh
Yeah,
man.
and it got rejected because it wasn't interesting, it wasn't important, and it wasn't scientifically rigorous or something like that. And I was like, okay. Uh, Fast forward 5 years, and it's one of the largest discussions we're
It's,
having,
I
right?
mean, it's the main discussion.
Right.
Like,
So whatever,
I'd
whatever.
say it's funny. So I have only been the chair of conference programming for the last 2 years, maybe a little
So
bit
it
more.
wasn't your fault. You
So it
didn't do
wasn't
anything,
me. And
and it
we
wasn't Kyle.
we've had some intense discussions about that in recent years, but it's great because most conferences you have once a year, even rigorous academic conferences, that's sort of how it is. In ACS, we have a spring and fall meeting, and they're both like big, rigorous academic
Well,
meetings.
yeah. I mean, it was hard for little old me to be up against, some of these, large labs and or, academic
Yeah,
facilities that had much larger studies, much more. I mean, I had no money, right?
my
And it was a small study with like 20 samples with a clear pattern, but it wasn't good enough. Whatever, it's fine. It's
it's
published
it,
now.
yeah, right. I mean, we could play with some of that FOIA data that Yasha collected
I know,
and
I know. Well, so
really.
let's talk about okay, so the last thing, and we got like 9 more minutes on the clock here. Let's talk about the importance of education.
The most important of
The
all of
most
them.
important.
I mean, education, communication, it's the core of really what makes us humans. Um, it is how we all learn and grow and what shapes our perceptions of the world and colors our instinctual reactions. And so for since 1937 or even before that, really, the primary narrative has been about cannabis being harmful and toxic and all these things, and the rigorous academic study was used to just drag cannabis and prove how it's bad for society and bad for people and bad blah 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 blah. And generation after generation, you form durable opinions that people don't even understand why they have them or what they're based on because they're not actually based on anything. They're just based off of, tradition, essentially. And legalization is the first step. It's like the prerequisite to the first step. We now have to actually rehabilitate the
Mm.
image and the understanding. And so there's room for a lot more science, a lot more science. We talked about nobody has decortication to do any of the fiber things or seed processing or whatever. Like that's because the system has been built for specific inputs and has been run like this for 90 or 100 years.
Mhm.
And without changing hearts and minds through not just research and publications, but communicating to the common person the benefits and the things they're missing out on and the trade offs that were made without their consent on petroleum being the input for everything in their life as opposed to a a biomatter feedstock like cannabis. It's hard, and you can't change systems until people that make up those systems want to change. And so education is the core of that. And that's why people care so much about where their kids go to school and what they're learning and, what books are in the library and all of that. And it's like they banned all the cannabis books, essentially. They banned them so thoroughly that we don't talk about them. I remember, I don't know, 20 years ago, like back in the Napster days, more than 20 years ago. Um,
Napster.
and like, yeah, and like coming across a file that was filled with like cannabis cultivation books and stuff like that. And being like, oh my God, I had no idea that any of these books even existed, let alone that I should be looking for them. And it was just like, it, that should not be how it is, right? Like
Right.
Jeff Lowenfeld's books should be everywhere.
Yep. Yep. So my parents are from Tasmania,
Okay.
incredibly isolated. They did not grow up in the hippie 60s era, even though it was the 60s. They had never even seen pot, smoked pot, nothing. Very anti-pot, anti-cannabis. Cannabis is bad, blah, blah, blah. I went through my first round of college, took every class on drugs and sex that I could find, even though my major was animal anatomy and physiology, which is real close. Um,
I
but
mean, sex is at the core of all of that.
but then, when Colorado legalized cannabis and I had finished my master's, I had done 2 years at the Denver Botanic Gardens on working on, conservation genetics. When Colorado legalized for rec, I saw a really good um, opportunity for me to go back to school and study this plant that had basically not been studied at all, at least for the last 100 years, and especially since genetic tools had been developed,
Yeah,
right?
right.
And so talking to my parents, telling them that I'm going to go back to do my PhD in cannabis was like, they're like, "What? Like you're gonna study drugs? Like it's and deconstructing everything that they thought about cannabis.
Oh my God.
I'm not going to be smoking pot. We're not going to be doing pot. We're not going to be growing like deconstruct.
No, it's just
It's just a
they didn't
plant,
study this. Think about all the citations, right? Like we
right?
could just
It's
make
just
them
a
all these
plant.
papers.
My argument is it's just a plant. It's been used for,
Tens of thousands of
debatably
years.
10,000 years. It makes all of these incredibly amazing compounds. Like it's arguably one of the most interesting plants in the world, one of the most recognizable plants in the world, one of the most utilized plants throughout history. And we know nothing about it virtually, like in terms of the plant. Like it's
Like
been,
there's this gaping, like a
yeah,
big book of like what we know about biochemistry and the world and plants, and it's like the huge chapters just been ripped out.
right. We know everything about corn and soy and cotton, and we know nothing about cannabis. And so that was my stepping in point is I'm not studying what smoking pot is like. I'm studying this plant and the genetics and the relationships of this plant that has gone relatively well in terms of population genetics. There was really hardly anything done at that point in time.
Yeah.
Um, I think Carl Hillig was one of the only sort of large population studies that had been done, and he had to kind of do it outside the US because hashtag the United States is
Yeah, right. Of course.
thick butt. like, but whatever. So it was really interesting because there wasn't like a whole lot of literature for me to dive into. Whereas, you start your PhD, it's like you gotta
Yeah,
start reading the literature. And I'm like, there is none. I had Clark
right. This is
Merlin
what
who,
I'm thinking as you're describing this. I'm like, you're going to school right as they're legalizing. Uh, what is the literature base?
right.
Like,
And so like I'm citing things like Danny Danko's book and my PI is like,
veggie
you
cannoli.
can't cite that. And I'm like, there's nothing to cite. Like there's no like the pool of literature is very small. Like I'm gonna have to cite some of these cultivators that have written books or written blogs or like
How
they're
many citations does your thesis have?
so my dissertation has I think 200.
Okay.
But it does include a few websites like Leafly Seed Finder, WikiLeaf. It does have books like Clark and Merlin, like Jorge Cervantes, Marijuana Grow Bible. Like
I
it's,
mean, like
but
Jorge Cervantes is a classic, right? Like it's
yeah,
foundational.
yeah. And and just because it hasn't been peer reviewed, I mean actually if you read the list of people who contributed to that book, it's in the
Kind of
front.
has been peer reviewed.
There is a long list of really respectable, knowledgeable, educated folks that contributed to that book in, one way, shape, or another. So, it was just funny because I had to come at it from a completely different
Yeah.
direction that most people come at their PhDs from. So, anyway, I also agree with you. I think education is fundamental in deconstructing some of these ideas that have been so ingrained in our culture for so long. And, I do teach cannabis science courses. I think every day when we speak to people, we're educating people. Um, and I guess I know you have to go and pick up your kiddo. So, uh, tell people how to find you. You're on LinkedIn for
You
sure.
definitely on LinkedIn. Uh, you can come to our websites. You can email me, Evan at scientificcell.com. Yeah. Uh, call me at the office. Like,
All right, I'll put up some of your information at
yeah,
the end of
please
this so
do.
that people can get a hold of you if they have questions about standards or
Sta-
sex tests or
Standards,
the Purple Pro.
chemical analysis, uh, efficiency. Yeah,
And you're
if
very
you're,
generous with your time and your knowledge, if which is always nice to meet people who are not, it's not going to cost you $100 to pick your brain. Like you are willing to answer questions and help people who need
always,
help. So
always. I mean, people have been generous with me over the years. I ask a lot of questions, and so I mean, if I can answer a question, I want to. So
good, excellent. Well, thank you so much for your time, Evan. It's always a treasure to talk to you. I know we'll probably see each other here in the next few months, maybe at MJ
I
Impact.
hope so.
Atlantic City, I'm planning on going. I hope you're planning on going too. So until then,
That sounds great.
hasta la
Thank you so
pasta.
much for having me. This was wonderful. I really appreciate it, and I can't wait for it to come out.
Awesome. Thank you so much, Evan. I'll talk to you
Thanks.
later.